Hello and welcome to a short video describing the ACS course Pharmacokinetics for Chemists. My name is Terry Kanakin and I am the instructor for this course. I currently am Professor of Pharmacology at the UNC School of Medicine, Chapel Hill, North Carolina, but I've spent over 32 years in drug industry, 7 at Burroughs Wellcome and 25 at GlaxoSmithKline. I present the course from the perspective of chemists and biologists progressing new molecules forward towards drug candidate selection. For the year 2018, the course is presented in 3 cities throughout the year, San Diego, Boston, and San Francisco, and 4 times on the web as a webinar in February, April, October, and July. Details of these offerings can be found on the ACS website shown below. Irrespective of what drug target or therapeutic area is of interest in a discovery program, a common bottleneck in the process is found in ADME, an acronym for Pharmacokinetics and Safety Studies. Thus, all new drugs must be able to enter the body, distribute to the therapeutic target organ, remain there long enough to cause response, and cause no harm. The course content is based on the answer to 4 basic questions to describe therapeutic drug action. Of any dosage given, how much of the drug is absorbed into the body? Once absorbed, where does the drug go in the body? Once the drug is distributed, how long does the drug stay in the body? And finally, how often must the drug be administered to attain a therapeutic steady-state drug level? The impact of pharmacokinetics has changed radically over the past 15 years. In 1991, 48% of all new drug candidates failed because of inadequate pharmacokinetics. By the year 2000, this was reduced to 12%. Until now, it is less than 1%. There is no excuse for discovery programs to lose a new drug candidate because of inadequate pharmacokinetics. A number of factors have contributed to this advance in pharmacokinetic development. Perhaps the most important being the availability of new, inexpensive, and predictive in vitro PK assays that can now be used to eliminate problem molecules from progression paths. This course focuses on the application of these new assays to new drug development. These assays will be described in terms of what question is the assay designed to answer, how is the assay done, what data emerges from the assay, and what sort of numbers would you wish to see coming out of the assay. And finally, the interpretation of the in vitro PK data in terms of the development profile. By the end of this course, you will have a feeling for the type of data common to robust drugs in the clinic, and also the assays that are essential to providing these profiles. A well-known fact about new drug discovery and development is that often a large number of iterations are needed to attain the proper balance of activities. Basically, 3 separate structure-activity relationships must be satisfied: primary activity, ADME, and safety profiles. An important theme throughout this course is that these structure-activity relationships can be completely independent of each other. This makes for a complex process. For example, this scaffold for the blockade of IGF-1 receptor has a safety issue in the inhibition of CYP3A4 and CYP1A2. However, it can be seen that as chemists modify the R group, They are able to reduce the safety hazard while keeping the primary activity unchanged. This concept will be applied in the course to show how ADME properties can be changed through medicinal chemistry. By the end of the course, my aim is to give you a sense of pharmacokinetics and what it means to the discovery process. We will work with the two main pillars of pharmacokinetics that essentially control all aspects of in vivo PK, namely clearance and volume of distribution. These concepts will be discussed in terms of in vitro assessments of clearance and volume of distribution scaled to total body in vivo PK. The course will first define the important processes involved in the absorption, distribution, and excretion of drugs in vivo. Thus, the drug is absorbed into what is called the central compartment. This is where we sample in vivo concentrations and is usually the systemic circulation. Although it is called free drug here, the drug will not remain free in this compartment long, but rather will quickly bind to plasma proteins. It may be degraded in this compartment by enzymes, especially if it is an ester, and if polar, it may be excreted by the renal system the minute it enters the central compartment. The drug will distribute throughout the body and may concentrate in various parts of the body, such as adipose tissue. The hepatic system is designed to protect us from foreign chemicals, and our drug is a foreign xenobiotic. The liver will convert the drug to an inactive, or at least a more polar product, and these polar products will be excreted by the kidney. And if we are fortunate, a fraction of the dosage will find its way to the desired organ of choice. So one way to view this process is to consider our therapeutic organ as competing for drug in a distribution system. This course will consider techniques to maximize delivery to the tissues of choice. Discussion of development will be done within a greater context of the drug discovery process. As a starting point, theoretical and practical aspects of the main components of development will begin the course. Ideas such as drug-like activity, absorption, distribution, metabolism, and excretion. These ideas will then be put together into a discussion on in vivo pharmacokinetics. Real examples from the literature will be used throughout the course to illustrate concepts and techniques. Practical issues will also be discussed, such as the role of PK in any discovery project. Within any new drug discovery project remit, there will be certain ground rules, and this course will talk about how PK issues affect those ground rules. Specifically, how can pharmacokinetic help a project get to where it needs to go? In this example, there are two requirements of the proposed new molecule that involve PK, active by the oral route and once-a-day dosing. Each can be assessed in candidate molecules. Thus, permeation and studies of drug stability can furnish predictions about oral activity, and measurements of half-time can provide guidance for dosing regimen. The course looks at practical ways to modify PK. For example, since half-time is a ratio of volume of distribution and clearance, these two activities may be adjusted to attain favorable half-times for dosing. An example is shown here. In essence, the project team is aiming for a strip out along a surface to achieve a once-per-day dosage regimen. One way is to adjust clearance. In this example, it is known that the hepatic system attacks the methoxy group of metoprolol. Conversion to pentaxolol eliminates this liability. This produces a 61% decrease in clearance and subsequently produces a half-time of 17 hours suitable for once per day dosing. Other strategies may involve modification of the drug-like properties of the molecule. Remember that ADME properties may be modified without changing primary activity. In this example, the log P value of the molecule is known to be correlated with clearance. Addition of the hydroxyl group, as shown, reduces log P and takes advantage of the correlation of log P and clearance to reduce the clearance of the molecule. A different strategy may be to change the volume of distribution of the molecule. In this case, advantage may be taken of the fact that pKa is correlated with volume of distribution. For example, in the case of the weak acid nifedipine, the volume of distribution is low with a correspondingly low half-time. In contrast, the basic molecule amlodipine has a much higher volume of distribution. This increased volume of distribution leads to a much longer half-time suitable for once per day dosing. This strategy was used to convert the short half-time antibiotic erythromycin to the once-a-day drug azithromycin. In this case, a second amino group was introduced into the molecule to increase pKa. This led to a 35-fold increase in the volume of distribution and a subsequent 34.5-fold increase in the half-time. The course also considers FDA requirements for drug approval. The main considerations here are safety and issues such as drug-drug interactions. The various ways that provide these data will be discussed in the course. This will be put in the context of the FDA guidelines for development. Some PK/PD modeling at this stage also will be discussed, such as the FDA suggested application of the static or the net effect model for drug-drug interactions. The course concludes with a discussion of safety pharmacology as it relates to PK and pharmacodynamics. Inevitably, safety issues will arise, and the course aims to put these into perspective in terms of weighing the balance for FDA approval. Discussion will center on assessing safety, identifying hazard, defining risk, and the use of early-stage safety assays. The process of safety assessment will also be covered, along with some special key topics. The most common safety issue leading to cessation of development is hepatotoxicity. This is because the liver usually gets the highest dosage, and either dose-related or idiosyncratic toxic effects are seen at this stage. Another key issue is blockade of the hERG potassium channel, leading to catastrophic lethal cardiac arrhythmia. Finally, we will discuss cytotoxicity and mutagenicity. The course also comes with course materials. You will receive a complete color set of printed slides. For the webinars, you'll have access to the recorded lectures. There's supplemental reading material, problem sets, and answers. I also offer online support. After the course is finished with, I've had students send me questions and emails months after the course, and I welcome this. You'll also get electronic copies of my 2 textbooks on pharmacology. So you will get electronic copies of a pharmacology primer and also pharmacology and drug discovery and development. So that takes us to the end of this video. You can find more course descriptions at the URL shown there, and I'd be happy to answer any questions you may have. Just send me an email. Thank you for your interest and attention.